anchors is named Becky Quick,
Mm-hmm.
and she just announced that her daughter has SYNGAP.
Oh, wow.
Yes, and it's amazing because she has such a strong um podium. Like she is like the most incredible her position in life of interviewing companies and dispensing market news and going to Davio. I'm drawing a blank of what's in Davio every year. Big stuff though. They're having the first ever Cure Summit sponsored by CNBC in a couple of weeks in New York City. So I am super excited to attend,
No, that's awesome.
isn't it?
Yeah.
Hi, everybody. Hi, Matt. Hi, Paul. Hi, Andrew and Kirill.
Good morning.
Hi,
Hello.
Amber.
Good morning.
Hello, everyone. Arvind here.
Okay, do we see Maria on? Because she's a pretty important person, being that she's presenting. Hi, Mike. Oh, there's Maria.
Hello.
I am making you co-host just in case
Yeah,
we have
Hi.
tech problems.
thank you.
I don't know if everyone follows us on social media, but I have nothing but good news to share as far as it goes for SLC6A1. We have a $750,000 matching grant, but we don't get that money until we raise it, and we're already halfway there on February 12th.
Oh, great. Congrats.
I know.
Good
And then
job.
Sergey Brin, the founder of Google, has an organization similar to like Chan Zuckerberg, and they are giving ARIA grants to centers of excellence to focus on very select few genes. And when I say very select, and none of it is public yet, but I have a lot of reasons for optimism, and I can confirm that one of our centers of excellence is receiving a $15 million grant to build infrastructure to advance precision treatments and build like amazing data sets. And super excited. It just might be one of a gene we're all on here for.
That's wonderful.
Fantastic.
Great news.
That's fantastic. Great news.
I know. I mean, it's felt like you guys understand it's such a grind day to day. And then when all of the good news starts happening, you're just so excited. I hired the K-pop Demon Hunters to come and perform for Maxwell and Riley's birthday party. And I invited both of their third grade classrooms. It's going to be like, think Old School, that movie Old School with Will Ferrell, except for 8-year-olds.
That's great. Yeah,
Oh,
rare disease is really, I mean, like even just kind of keeping a little bit of an eye on things. It's amazing how much is in the public going in the public eye. Like Becky Quick, probably.
we
Yeah,
were just talking about that, Mike. I'm going to the conference in 2 weeks.
great, great. Yeah, that's fantastic.
We just need people to get diagnosed, as sick as that sounds.
Yeah.
Okay, everybody, we can get started. Thank you, everybody, for being at our February virtual lab meeting. It is February, so my dad joke of the month is, will you be my co-Valentine? We have a lot of newcomers every single call, and it is simply amazing. So, as a crash course for these, I'm Amber Freed. I founded SLC, and we are a patient advocacy group dedicated to improving the lives of children and families affected by SLC, and we do that by raising awareness and fundraising to ultimately advance research that will result in a cure. We keep a list of contact information. I'll put a link in the chat. If you are not on it, it would be great if you could add yourself. And also, if you are new here, would you like to take a moment and just give a super quick introduction?
Hey everyone, it's my first time. My name's David Olson. I'm out of Dallas, Texas at UT Southwestern. Was working with Kim Goodspeed before she left, and so now in a position to start to help with the natural history study for SLC6A1.
Amazing. Would anybody else like to speak? And the document that I put in the chat has contact information, so feel free to open it and scroll through to see the other participants. And there's also contact information, so if you'd like to follow up with somebody specifically after the call, please do. Our symposium was incredible. We're already planning next year. I simply can't say enough about the amount of momentum we have. We could employ a staff of 10 people right now working full time, but unfortunately that money is better served writing grants. So rather than do that, it's me. And so that's why sometimes my emails are delayed. We have a lot of conferences coming up, and I would love for anybody to attend. I will be spending Rare Disease Day at Notre Dame. There is a family named the Reisenhauers that actually started a program to get a minor in rare disease. Advocacy, it kind of covers the full gamut, and a lot of pre-med students are working on this minor. So the entire minor program is going to be in attendance. The documentary Too Rare to Care will be playing. I will sit on a panel afterward. Anybody that would like to attend, we would love to have you. We were talking earlier that one of the CNBC anchors named Becky Quick recently announced her daughter has SYNGAP. And she has such an amazing podium. She knows all the biotechs, like they legitimately come on her show daily. And she has come out swinging in a big way. There's going to be the first CNBC Cure Summit in early March. I will be in attendance, trying to think of the next couple conferences. But unfortunately, my brain goes out 1 month. More things are, Terry Joe will see me in New York. There are other things coming up, a couple things in Boston. I'll put that on the email I send out. So symposium content, we're still making sure we have permission to post everybody's presentations. They're all going to be available on a Google Share Drive, as well as a professional B-roll, and we have some great pictures of everybody. I can just tell you they're probably gram-worthy, so just plan to have a new Instagram profile pic here shortly. As everybody knows, Maxwell received gene therapy in September, and everybody is always asking. He is doing fantastic. We have filed for a patient-focused drug development meeting with the FDA. We've made a large investment in regulatory this year so that all of those regulatory hurdles that give you a migraine just thinking about will be in place. Now that we've de-risked the transgene and we've repurposed a drug, we want to make it even easier for anyone that's interested in us to be successful. So now the FDA will be very, very familiar with us. And just to take a moment to acknowledge what a crazy time point we are in history, that typically you schedule these meetings to educate the FDA about the disease, to make drug development easier. Well, we're going to the FDA and saying, hey, we have a gene therapy, we've repurposed a drug. You already know us from paper, but thought it'd be cool to put a face with a name. Like, we took the opposite way. And that just speaks volumes to what a patient organization can achieve. Rare Disease Day is coming up, and we have some great swag. Let me put the link in the chat. It is free. I think most people have ordered their swag, but if you want to be the envy of your friends, I would go ahead and put your order in for Swiftie bracelets. There are some funding opportunities coming up. We just had SLC represented at Uplifting Athletes. Caddy, is Melissa on the phone? Or did you want to say anything about the night? A member of Caddy's lab got a grant. Caddy, I think you're on, but if you're trying to talk, just write in the chat.
Is she
Jen,
on? I didn't see her in the list of people, but maybe I'm wrong.
I thought I saw her, but when you're presenting
Hi,
your
Amber, it took me. I think I have some problem. Yeah, you are frozen. Yeah, you're frozen. I cannot hear it from you. Yeah, yeah, thank you. Yeah, so we're very excited for the grant, Melissa. Yeah, she did a wonderful job for we for the grant. Basically, the grant is based on her work, sister's work. She studied 32 variants. She compared for phenylbutyrate and butyrate and multiple other small molecules. The paper is in the final stage of revision. We'll hope to see online very soon. Yeah, thank you so much, Amber.
Yes, of course.
Yeah.
We're just so excited to celebrate every single person in this community. So the next upcoming meetings I'm very excited about, and we're taking a different path in our traditional meetings. I think one of the most impactful sessions at the symposium was when we were talking about behavior. And we talk about seizures a lot because that's measurable and it's super easy to see on an EEG. But when you talk to the parents, behavior is different. And reading about it in a paper is much different than seeing kids drive around in restrained car seats and what the day in, day out looks like. And so we've decided to take this very, very vulnerable approach to the next couple meetings. In March, we are going to have parents present about some of the specific things that have happened to them. The meeting won't be recorded. We won't allow note takers because this is so much shame. Like our kids are getting kicked out of school. I'm on private chats with our parents that for the ones that are cognitively able to connect the dots, like parents have privately shared that they're afraid their kids could be the next school shooter. It's just a terrible place to be. And one of the things we've privately been talking about is we now have the ability to extend our children's lives and maybe calm the seizures, but the behavior, there's just no answer for. And this is such a dark thing to say, but ponder this. If you were holding your newborn baby and you had the choice to have a couple of really good years with them and knowing that they were going to pass away, or survive into adulthood and complete an atrocity that, I don't even want to say out loud, what would you choose? And I think that in our darkest moments, that's what a lot of SLC patients are facing. And so the next meeting, parents are going to be very, very honest and talk about some of those moments and things that they're probably not even sharing with schools and friends because they're afraid of retribution. That will be March. In April, we are going to present the scientific side, and a group of scientists will be talking about the link between SLC6A1 and schizophrenia that he found in his research. It will be a different day of the month than we usually do because of the Easter holiday. Oh, and this slide actually is wrong. Then the following meeting will be our clinicians talking about some of the behavior and what can be done and what can we change today? What do we need to think about clinically as it goes into science and what to study? So I would say the next 3 meetings will be very important and interesting. We will tape the scientific and clinical viewpoint, since that won't be anything identifiable patient-wise. Lastly, does anybody have any meetings or any announcements they would like to make?
Yeah, I have an announcement that earlier this week I submitted an R01 for natural history that includes SLC6A1. So fingers crossed.
Well, Sarah, thank you.
Congratulations to Sarah.
Thanks. Yeah, Amber, of course I was going to include you guys.
And now I will stop sharing. Oh, geez. Was I sharing? Why don't I see stop share? There we go. I will stop sharing and hand over to Maria Carmen Varela, and she is going to talk about her brain organoids. Maria, we are so excited to have you.
Thank you so much. Let me see if I can share here. Okay, 1 second. Okay, can you guys see that properly? 1 second.
Looks
Can you hear
good.
me? Okay.
Seems a little
Sorry.
big.
Yeah, I don't know why it's doing that. I hope it doesn't cut off the. Well, we'll roll with it. Okay, so if you guys came to the SLC6A1 Connect Symposium or saw my talk at AES, some of this might look familiar. I'm going to kind of review some of that, and then I promise I'll have some new, never before seen data at the end that I think will be exciting. So yeah, my name's Carmen. I working on SLC6A1 for quite a while now. And so we developed an organoid model that we think will work really well for testing or any kind of drug testing or disease modeling of SLC6A1. And so I'm going to tell you a little bit more about that and talk about the medial ganglionic eminence, just kind of for background, introducing our single rosette organoids, and then briefly touch on how we generated the MGE-like organoids, and then how we model SLC6A1 and some of the migration deficits and rescue that we found, and then some future applications in disease modeling for DEE-related disorders. So the medial ganglionic eminence, if you're not familiar in development, is this transient structure. Let me see if I can find my pointer here. Oops. Sorry, I'm trying to find my pointer. Well, okay. Can you guys see this little pointer? I think that's.
Yeah. It's lagging around though.
It went away. Okay. So anyways, I will just try to roll with it. This is a section of the mouse brain here that you can see the medial ganglionic eminence is this small area in the middle. And it's a transient structure. It doesn't last for very long, and the cells will migrate out of it up into the cortex, where they will find their long-term homes. And, sorry. Oh, there we go. And the medial ganglionic eminence is kind of the birthplace, the origin of many really important interneuron subtypes, including parvalbumin and somatostatin. Those two are the main subtypes that arise from the MGE. And the reason why these interneurons are really important, especially the PV, so I'm going to kind of focus on PV for most of this, but it's been implicated in epilepsy, intellectual disability, autism spectrum disorder, Alzheimer's, schizophrenia, and ADHD as well. So these interneurons, when they're dysfunctional, can cause a whole slew of problems. And so we're really interested in being able to model these in a dish. And one of the things that has been really hard is to produce parvalbumin interneurons in vitro. So that was the main goal of this project, was to create a medial ganglionic eminence organoid that was specific enough towards the medial ganglionic eminence identity to actually produce parvalbumin interneurons. So just kind of talking about why we chose the single rosette organoid as a platform for our disease modeling. You can see kind of there are two main flavors of organoids that are generally used right now. The first one is a single rosette, which just has one central lumen. And then there's the other more widely used type of organoid, which is a multi-rosette organoid that has many different lumens or rosettes inside. And with this multi-rosette kind of structure, you get a lot more variability. So just because of that, we decided to use the single rosette model for ours. So with our protocol, we kind of plate as a monolayer first, and then we start patterning that way, and then we cut them and plate them onto ECM, which allows them to really beautifully self-organize into the single rosette structure, and then we grow them in suspension. And so this method was developed by Andrew Tidball, who was previously a postdoc in our lab, and he developed the cortical version of this. And so we kind of use this as a base for our medial ganglionic eminence organoid. And this is just kind of a larger overview of this project, but we tested a lot of different signaling molecules, different pathways, timing, concentration of all of these different patterning factors to determine which could give us the best MGE-like organoid, and compared it with human fetal development, characterized all the different interneuron subtypes, and then characterized the migration of them as well, and then did some EFAs, and then showed in disease modeling with SLC6A1 that it can be useful for that as well. So just to kind of confirm that we actually do have an MGE-like identity, we would like to see some of these key markers that are commonly seen. FOXG1 is a forebrain marker. SLC6A1 is kind of the most specific MGE marker that you can have. And then NKX2.1 is also expressed in the MGE, but in humans, it can also be expressed in other areas like the hypothalamus and POA. And then we don't want to see PAX6 because that's a marker of the cortex. And we don't want to see RAX, which is a hypothalamic marker. So just confirm in our day 30 MGEs with single cell RNA sequencing, we can see we have really strong expression of these important markers from the MGE. And then we also have GABAergic markers as well. And then to confirm that we don't have off-target, we have very minimal expression of some of these off-target markers from different surrounding regions. And then this is just immunostaining of our MGOs at day 30 and then day 90 here, just showing the progression of these markers as GABA starts to be expressed a little bit around day 30 and then continues to expand and be expressed largely in the organoid, as well as LHX6 and NKX 2.1. It does go down a little bit in the cortical migrating populations. So this is what we would actually want to see. And then OLIG2 is an oligodendrocyte precursor marker, but it's also expressed in the progenitors, the interneuron progenitors from the MGE. And then this is just comparing our 3-month-old MGOs with a published data set, Shi et al., that took human ganglionic eminence dissections and did single cell RNA sequencing on them. And so we were able to show that our MGOs here in yellow really nicely co-localize with the Shi et al. MGE, which is shown here in blue. And so we were really interested in pervalbumin. And so we did kind of a time course to see when pervalbumin comes up. And here at about 3 months, we do see about 1 to 3% of cells are expressing pervalbumin strongly. We actually do see sometimes as early as 2 months. But as we reach the 4-month mark, we see a really robust increase in pervalbumin. And that is maintained at about 30 to 40% of the organoid throughout the duration of the organoid's life. And then to confirm that this is actually really PV from pervalbumin interneurons, we co-stained with a marker for perineuronal nets, which is something that are kind of seen specifically around pervalbumin cells, and that's what's labeled here with WFA. So we can see some really nice co-localization of those two as well. And then just for one more confirmation, we looked at potassium channel Kb3.1, which is highly expressed in pervalbumin interneurons. So we can see at 3 months, we don't have it yet. We do have mRNA expression, but the protein is not there yet at 3 months. But by 8 months, we do see really nice expression of Kb3.1, which co-localizes with MEF2C, which is another marker that's co-expressed in pervalbumin cells. And then just to kind of go a little further, we wanted to see, there are 2 basic main subtypes of pervalbumin as the basket cell and the axoaxonic cell. And the basket cell tends to contact along the soma of the neuron, the target neuron, and the axoaxonic cell tends to contact onto the axon of the target neuron. And so we show here that we can see synaptic puncta with VGAT and Gefrin, which is a presynaptic and postsynaptic markers, onto our pseudo-colored excitatory neuron that has been labeled with cacti tomato. And we have the puncta here, and you can see them, they're co-localized really nicely. So this tells us that we do have basket cells. And then we also wanted to look for axoaxonic cells, these are really hard to generate. I don't think anyone has generated them in vitro before, but we show here with anchorin G, which stains the axon initial segment where these cells tend to target, and we can see some nice synaptic markers here along the axoaxonic section. So that tells us that we have both of the main types of interneurons that are made from, of the PV interneurons that are made from the MGE. And so we wanted to kind of characterize the electrophysiology of these organoids. So we fused our, MGOs with cacti tomato labeled cortical saucer, a single rosette organoid. And we labeled this with MDLX-GFP, which labels the young migrating neurons. And we put them on an MEA plate and kind of looked at the activity over time. And so at this time point in the early, it takes organoids pretty long to mature with really robust network activity. So at DIV 140, we see that they're just barely starting to kind of get some synchronous events here, but mostly it's a little bit of kind of sporadic activity. And then by day 210, you see some really nice spontaneous electric network bursts here that are synchronous and very rhythmic in nature. And then as you let the organoids mature even more to day 235, we see some really complex bursting activity that kind of varies in its characteristics. And so we added some bicuculane to make sure to see if the GABAergic system is functioning in these organoids. And we can see with the application of bicuculane, we have an increase in the mean firing rate, which bicuculane blocks the GABAergic signals. So this tells us that with this blockage, we have increase in activity, which lets us know that our GABA system is working in these organoids. And then just to kind of do some spike sorting to characterize the different types of units that are being picked up by our MEA. We have a really talented postdoc that joined the lab recently who has done this in animal models. And so he did some spike sorting from our MEA data and kind of looked at during a burst what the different cells characteristics look like. So for this particular one channel, he just showing as an example, we have one cell that's kind of characterized as a regular spiking cell. And then we have two that are characterized as fast spiking cells. And we kind of used more on the conservative end of characterizing the fast spiking cells as having a trough to peak right here, duration of less than 0.4 milliseconds. So we have a very large population of fast spiking cells, and we think that we have so many, or we're picking up so many of these fast spiking cells because if you remember our image before, most of our parvalbumin cells are located around the edge of the organoid, and that is the part that is touching the electrode. So I think we're oversampling a little bit with the number of fast spiking units that we're picking up, but it definitely lets us know that we have some nice fast spiking units in our organoids. And then now I'm going to shift a little bit to show you how I've used this with SLC. So just kind of starting off with 1 month and 3 month MGOs, we wanted to check if SLC6A1 was highly expressed in our organoids, and it was, which is really great because I think other organoid models have had a little hard time with expressing SLC6A1. And so just to start off on my project, I made these CRISPR gene edited and control lines, as well as blood-derived iPSC lines. And sorry, my son is in the background. So I have a PBM. I'm sorry?
Can I jump in with a question?
Yeah, sure.
So Maria, I mean, you've probably covered, I don't know, 5 or 10 years of research in 10 or 15 minutes. Pretty impressive.
Yeah. Yeah.
So just to summarize, I guess what you've said so far is by taking human fibroblast biopsies, you can create organoids from them that model the median ganglionic eminence derived cell population that now is found in the striatum. So you're looking at GABAergic interneurons with this model as a model system. Is that right?
Yeah. So the medial ganglionic eminence has more than two, but generally two main populations that come out of it. It has one early migrating that migrates down to the striatum and subpallium regions, globus pallidus, and then it has another population that migrates up into the cortex and kind of fills in there. And so if you can see on this image right here, this larger part on the bottom here at the 3-month mark, these are all cortical destined interneurons, and then these clusters up here are the more subpallial ones. So we do make all of the subtypes or kind of lineages that the MGE makes. So if you're interested in striatum or other subpallial regions like globus pallidus, we also have parvalbumin cells that kind of tend to look more like this area.
And yeah, that's great that you refer to that map there, because that was my next question. I think you said earlier that the SLC6A1 is the definitive marker for GABAergic neurons. Is that right? And if so, are there cell types that just don't express it but are also present in the organoid?
So yeah, it is expressed on all interneurons, on all GABAergic neurons. It's more highly expressed on parvalbumin interneurons, and I think some other interneurons like LAMP5, which come from a different area like CGE. So it is VIP, those kind of cells, they express them a little bit higher, express SLC higher, but from the medial ganglionic eminence organoids or region, between provalbumin and somatostatin, which are the two main subtypes, provalbumin expresses SLC6A1 a lot higher than somatostatin. And we do have somatostatin in our organoids as well. So we can kind of like look at the expression that way.
Great, thank you.
Okay, so I kind of made some of these lines, both from skin fibroblasts and from patient-derived blood, and then have the isogenic control and created MGOs from that. And this is just kind of giving a little bit more information about my CRISPR lines and where I targeted them. So in case you're interested in the protein, so this is a known mutation that results in a stop codon in this region right here. And so I wanted to target this because this is known to be pathogenic, and it's kind of guaranteed to have a truncation at this spot. So I kind of made my guide RNAs to try to reach this area, to specifically cut at this area. And so I have right here the different mutations from the HETs 1 and 2 line and knockout 1 and 2 lines. These are compound heterozygous, so both alleles are mutated. And then the blue here is just showing the actual truncation sites or predicted truncation sites of the protein as a result from these mutations. And then we have 2 patient lines here that I've been working with. The data in this talk will be mostly from this one right here. And that's also a stop codon. Actually, just to confirm, all of these, all of the mutations that I'm working with, or the variants that I'm working with, are truncation variants. So you can take that with a grain of salt, because I know that there are a lot of missense variants as well. So that might be there might be something that I might see with these truncation variants that might not be so obvious in missense variants. And so just to confirm that GAT1 is reduced, this is mRNA expression from RT-qPCR, and we can see in all our HETs and knockout lines, we have very low expression. And this is the patient line, which even though the patient is a HET, it has very low expression. So we're not really sure why it has such low amounts of expression of GAT1 mRNA. And so this was something that was kind of an accident that I noticed about the organoids. So we plate these on thick extracellular matrix when we're growing them on day 4. And then usually we pick them off about day 10 and then grow them in suspension. But if you leave them on, the neurons like to migrate out, or the neuroblasts like to migrate out into the gel tracts. And so if you leave them on long enough, you'll see a lot of migration come out by day 21. And so in our control and our wild type, so the wild type is the blood derived and the isogenic control is the CRISPR derived. And in those conditions, we see a lot of cell migration coming out from the organoids. But when I looked at the HETs, the knockouts, and the patients, there was very limited migration coming out from these organoids. And so I kind of continued with this line of investigation. And this is just the quantification, because I was curious about processy outgrowth or cell body migration or nuclear kinesis, because that's something, those 2 things are characteristics of these migrating cells. And in these PET conditions and knockout, we see a reduction in both the processy outgrowth and the cell body migration. And so we wanted to test, is this migration phenotype really linked to GAT1? Because it's not something that, to my knowledge, has been reported for SLC6A1. So we wanted to add in, back in the wild type GAT1, to see if it rescues the phenotype. So we have these doxycycline-induced wild type SLC6A1 here, these vectors that we put into the knockout line, iPSC lines, and then we made organoids from those and treated with docs with 2 different conditions. So initially I was curious if just jump starting them with the wild type addition of wild type SLC6A1 for a short period of time would kind of get them moving. Or I was also wanting to know, does it require SLC6A1 for the entire migratory period in order for them to continue migrating? So I did 2 conditions. One was only on for a few days, and then the other one stayed on until the terminal time point at day 21. And so this is the wild type stained with Th1, which is a marker, neuronal marker, and DNA here, which lets you see the cell bodies. I pseudo-colored it here with white so you can see some of the puncta a little better. And then this is the knockout. I just labeled it as KOI, which is just the inducible knockout. And so you can see here much less processing extension and migration of the cell bodies out into the ECM. And this is without DOX. And then this is with DOX, which initiates the expression of the wild type SLC6A1. And you can see a really robust increase in the migration. And this is the cell bodies here that have migrated out. And this is quantified here in both processing outgrowth and cell body migration. And you can see, especially with the processing outgrowth here, it even surpasses the wild types processing outgrowth as well. So this is just kind of showing the time condition. So we found that you do have to have the wild type SLC6A1 expressed through the entire migration period. Otherwise, they'll just migrate for a little bit and then they'll stop as soon as the wild type is not expressed anymore. And you can see that in some of these right here, where it's significant, but not a full rescue, just a partial rescue. Okay, and so then we wanted to see, well, does this migration phenotype hold true in a 3D, more kind of physiological condition? So we wanted to fuse them and make assembloids with the cortical organoids. And so we fused them around day 56, and then we imaged them for the migration. And this is just showing the different lines, the wild type and the isogenic control. After 2 weeks of fusion, you see a lot of cells have moved over into the cortical region, which is labeled in red. And then in the HETs and the patient line, we see very few cells that have crossed over to the cortical side. And it does seem that they kind of congregate or gather around the boundary of the cortical. So they seem to have some trouble entering the or crossing the cortical boundary. And this is quantified right here. Okay, and so then we wanted to ask, is this migration deficit cell autonomous or non-cell autonomous? So is it something that is happening intrinsically inside the cells, or is it something that's being influenced by the surroundings? And so we did a mix and match with the fusion, so mixing and matching the different genotypes for the cortical organoid and the MGOs. And in the conditions that have the wild type MGOs, you can see here and here, the 2 greens, that a lot of cells have migrated over. And then in the conditions that have the patient ventral, which are the 2 blue conditions here, you can see even though they're paired with a patient cortical organoid or a healthy wild-type organoid, they still have trouble migrating into the cortical region. So this tells us that it's a cell autonomous phenotype that we're noticing here. And so this was something that kind of I found also by accident. So I was looking, kind of trying to understand what the mechanism of this might be. And I found a wealth of papers in the oncology field that actually do look at SLC6A1, because the expression of SLC, or the overexpression, I guess, of SLC6A1 is seen in a lot of cancer types. And so they found that the increased expression of SLC6A1 in cancer cells increases the migration and metastasis and invasion of these cancer cells. And so they've done this study has done some knockout or knockdown of SLC6A1. And they found that with SLC6A1 knockdown, which is in the white condition here, you see a decrease in migration and a decrease in invasion. And then they've did a lot of experiments to determine this, but they determined that it's regulated through the PI3K-AKT pathway in these cancer cells. And so they use this AKT pathway activator, and by increasing the activation of AKT pathway, or the phosphorylation of the AKT pathway, they rescued, or they increased the migration and invasion again. So that does say that it's linked to SLC6A1, and it's also somehow regulated through the PI3K-AKT pathway. So I wanted to see if this still holds true in our organoid models and in neurons. So I just did some staining to check our phosphorylated AKT levels, and we actually do have less phospho-AKT. So I thought maybe we can try this drug as well on the migrating neurons and see if it, and the patient ones or the het ones, to see if it rescues the migration phenotype. So this is just really basic kind of outline of what I did. Was I did fusions at day 56, like I normally do. And then I gave them 45 days to just see what would happen. Could they still migrate? I wanted to give them time so that I can get a before and after drug. And this is kind of relevant because my cells that migrate from the MGE in humans continue to migrate well past birth, even up until age 3. So of course, if we're going to have any treatment that's regulates the migration of interneurons, it's going to be after birth, right? So I wanted to see if we kind of give them this drug after there's already been a little bit of delay in migration, is it enough to rescue the phenotype? So I gave them the drug 45 days after fusion, and then 2 months after fusion, I looked at it and I ended the drug, and then they continued to grow after that. Oh, so sorry about that. So this is 2 months post-fusion, and these are the control cells. Usually in our control organoids, we see a complete fusion or almost complete fusion by around 60 days, so 2 months. So in both of our control conditions, you see the cells have migrated and kind of filled out the cortical area already, and this is without the drug. And in the het conditions as well, we still see a reduced migration even after 2 months of fusion. So with the drug, really striking, it increases this migration by a lot, even after just 2 weeks with the drug. There's a really rapid increase in migration and ability to cross the cortical boundary. And then also I noticed a lot of much longer processes extension for these organoids. And so it seems to rescue some of this migration phenotype. And then since I didn't leave this drug on the entire time, I only left it on for about a month or 2 weeks to a month. It increased the migration during the treatment of the organoid with the drug. But then after that, the migration kind of slowed down after we stopped the drug back to its kind of regular level. So this is the kind of 100 days or 4 months post-fusion. You can see in the controls, there's, fully fused here, but in the hets and the patients, you still see some of the organoids. So the arrows are pointing to the MGO section that hasn't quite fully merged. And that was because I had stopped the drug much earlier. And so even though it hasn't fully merged, here's with the drug, sorry. This is with the drug. So it encourages it to merge with the organoid fully. However, some conditions, some organoids still have a small portion of the organoid, the MGOs, that are still showing even after 4 months. Oh, sorry. This is so we did MEA. So I was curious to see if if rescuing this migration would have an impact on the electrophysiological readouts, which we do see an increased hyperexcitability, increased bursting activity in the HETs and the patient lines. And so I took these organoids that had with drug exposure and without drug exposure, and I kind of pooled all the HET lines here together, and I pulled all of these as well from the HETs and the controls. So you can see in the control conditions, it doesn't affect the electrophysiological bursting patterns. It did a little bit in some of the organoids, it did decrease the migration. So I think in this one, you can see a little bit of a bud still left there from the MGO organoid, but it didn't affect the electrophysiological conditions. But in the HETs, which we see a great increase in number of bursting, it does bring it back down. The conditions that got the drug and increased migration brought the electrophysiological activity back down to normal wild type levels. So we think that this migration or increasing the migration is really crucial to getting these organoids to have kind of normal electrophysiological activity or network related activity. And I don't know 100% how long this would last. So these organoids are already over 200 days old, but if you continue to grow those, and we do see some kind of like seizure-like events, which the patient line especially has really long network bursts, so like 2 seconds in length, network burst duration, which is really long compared to our wild types, which are much shorter than that. So if that particular organoid maybe continues with this kind of seizure-like activity, we're not sure if the current interneurons that are there would be able to handle that. So that's something that maybe needs to be followed up in the future. So just in summary, we showed that we have this really nice MGE model, organoid model that can closely resemble and recapitulate the human MGE interneuron kind of development, and that they produce interneurons with high SLC6A1 expression, which actually are also expressed in our MGO astrocytes and oligodendrocytes, which I have an image later that I'll show. And then we show that we can model SLC6A1 and show a cell-autonomous interneuron migration deficit, which is fully rescued by the expression of the wild-type GAT1 and in the knockout MGOs, and that AKT activation rescues the migration and hyperexcitability phenotype in our MEA recordings. And really, the kind of crux of this is that we think impaired migration of the interneurons could lead to dysregulation of cortical networks, which we've modeled with our MEA, and it could contribute to the pathogenesis of SLC6A1 DEE. So we think this is a really useful tool, not only to model SLC6A1, but other DEEs related genes that are related to interneurons or astrocytes or oligodendrocytes even. And so it could also be a really useful drug for platform screening gene therapies or cell-based therapies. And just before I get to questions, I see Krill has his hand raised. This is just kind of showing in our MGOs a lot of the most common GABA-related or otherwise DE-related genes, and so we show with these kind of top ones here that we have really great expression in our neuronal clusters here as well. These are our glial clusters. We see some of these other ones are expressed more highly in the glial cluster as well. But all of this is our neuronal clusters here. These are the subpaleo clusters. So right here, if you're interested in any of these genes that are expressed along this region, which the GABA receptors are very highly expressed in these cells as well. This could also be a good source for modeling those genetic conditions. And then this is just kind of showing in our organoids the oligodendrocyte populations and the astrocyte populations that do express SLC6A1. And just to conclude, I'm really thankful for my undergrads who have really been stars in helping me with all this. I have 4 that have worked with me on these 2 projects, and my lab members, and of course my mentors. And I will, I guess I'll keep my screen on in case anybody has, well, okay, I'll go back in case anyone wants to see a slide. Hi, Kirill, you had a question?
Hey, Carmen, that was really cool.
Thank
It
you.
was great talking to you in December, and this was really interesting as well. I had a couple of questions. The first one maybe is something I missed, but for that cell autonomy experiment, so you're having an MG organoid, and then you have like a cortical organoid, and then you're trying to knock it out, knock SLC 6A1 out in either one of them and see where it matters. For the cortical organoid, how many of the cells are actually expressing? Because if they're mostly glutamatergic neurons, you expect they wouldn't. Is it the astrocyte? Do we have astrocytes in there? What's expressing it?
Yeah, we do have some astrocytes, not as much as in our MGOs. And then we do have some GABAergic cells in there as well. It's not purely glutamatergic, and those GABAergic cells are more characterized to be more like CGE-like, so they might have higher expressions of SLC because a lot of the interneuron subtypes that come from CGE have high expression of SLC as well.
Have you, in general, like taken a look at how much GAT1 would be expressed? For instance, like if you knock out GAT1 in MGE cells versus in the whatever cells you have in that cortical organoid, how in that final cortical organoid that impacts expression? Because maybe knocking it out in MGEs gets rid of the vast majority of it, which then kind of still raises some questions about cell autonomy.
Yeah.
I was just wondering, maybe.
Yeah, I do. I didn't show any of that here.
Oh, okay.
I do actually. So I have 2 of my undergrads are both doing senior thesis projects. One of them is characterizing all of the interneuron subtypes changes resulting from this in the MGOs, and the other one is doing that in the cortical organoids. So we definitely see some differences in the kind of subtype specification of the interneurons from the cortical organoids. Yes, so yeah, they do affect the cortical organoids as well.
And then with AKT, how specific do you think that is? Or you think that's just kind of, there's a mechanism that regulates motility and you're just driving that motility forward, not really in a mechanism specific way, but you're just, driving it.
Yeah, exactly. Yeah. So future applications would need to find a more kind of precise target for that. So it's the more I dig into the mechanism, the more complicated it gets. And I'm seeing that it's going to be a whole another somebody else's PhD project. So yeah, there's definitely things that can be done to kind of tease apart that mechanism a little bit more precisely. And basically the AKT drug is just, if there's any activation from the AKT pathway, kind of from SLC6A1, that drug just kind of overrides it and kind of pushes the downstream kind of applications of the AKT pathway. So yeah, basically it's kind of a sledgehammer just to see, we were kind of wanted to see if like increasing the migration would rescue the electrophysiological phenotype, the hyperexcited, which was kind of the terminal end goal of this. Like, is it that the cells don't have are reduced in GABA or in GAT1 that they have electrophysiological problems, or is it that they didn't get enough interneurons into the cortex? So yeah, we're somebody else will have to do that. Very detailed experiments.
Hi, Carmen.
One last quick question. Only as just one last one. I know you have a lot of other questions, but I know that you seem to have a pretty severe phenotype, and in the knockout, I assume migration would not be nearly as impacted, because if it was, I don't think the mouse would be viable. If you had to speculate, what do you think is different about your setup that impacts that migration so much more than it would be in vivo in a mouse?
Yeah, so it seems I don't know about migration in the mouse, but it seems that because we're able to have knockout mice and we don't have those in the human population. So I'm assuming in the human population, it's embryonic lethal. But in the mice, it seems that they're fine. So I'm not really sure if the mechanism is the same between mouse and humans for that. And I haven't characterized migration in mice at all. So yeah, so I'm not really sure. And we know that even in the mouse models that the knockouts, you know, have some of the phenotypes, but they don't recapitulate fully all the phenotypes of humans. So yeah, that's something that would have to be looked at a little more. But definitely in humans, I think it would be lethal. So that's probably why we don't see any of them in the population.
Thanks.
Hi, Carmen. My question is very simple. So, do you mention about in the heterozygotes the transcript, the mRNA is reduced, or maybe I missed?
Yeah. Yeah, I still don't have an answer for you for that. I don't know. I have had that question multiple times, and I'm not really sure how to explain that. But even in the patient, it has less mRNA expression than even some of our CRISPR heads. And I'm not
Like,
an expert on that
yeah.
Side of things.
Yeah, if you take a wild type as 100%, how much is the heterozygous and versus homozygous?
So it kind of varies. We actually, in our knockouts, we had one that was even higher than the heads, but that was because one of the alleles was terminated in an intronic region, and so that might have created some variability with splicing. So I think I showed it in the beginning, but we generally in the heads, it's about between 20 and like 20 and 50% reduction, or compared to wild type.
Oh, that's quite low. Yeah.
Yeah.
Okay, thank you.
Sorry, I had a question I wasn't able to raise.
I'm sorry. I think Henry has been waiting for a really long time with his hand.
I'll go after Henry.
Can I get to him first? Yeah.
Yeah, good morning. Thank you, Mario. This is great presentation, and congratulations on a lot of great data and very interesting model. My question there, I have two questions. One is that, so interneuron migration, at least in the mouse brain, is dependent on a KCC2 expression, the chloride homeostasis. And given that GAD1 in your model, there will be, I'm assuming, a lot of GABA spillover. And that alone also might change the KCC2 expression and NKCC1 switch. I wonder if you look at these two transport expression and maybe even more, whether you have done some patch clam to look at the chloride reversal potential.
Yes, that's a great question. And I did do that, yeah. So I actually have a whole other side of this project. Like, honestly, these two projects should have been two separate PhDs, but I do have another side of this project that looks at the maturation, cell division side of things. And I definitely do see changes in KCC2. So I see increases in KCC2 earlier on. So potentially changing the chloride homeostasis early on and giving them a more mature phenotype. So that's something as well that we're looking at. And I think I told Krill about that when I met.
Right. So if that's the case, that could align very well with why they're not migrating, because if the homeostasis already flipped, right? I mean, So in that case, would knocking down KCC2 might be a way to rescue?
Possibly. Yeah, I haven't tried that. That's a great idea. But yeah, I haven't even tried that. We did have actually a KCC2 knockout line that I was working with for a little bit, but I haven't done like double knockout, with SLC and KCC2 or double reduction. I guess you could use like a virus to reduce or something, like siRNA or something. You could probably do that. Yeah.
Because you probably don't want to knock out KCC2 all the time.
Right. Yeah, exactly. Because then you just cause more problems.
Right.
Yeah. Exactly.
My second question is related to the AKT pathway and PI3K. Downstream of that actually is mTOR, and that might be related to oncogenesis, and so some other overexpression is actually also a problem. So in your drug rescue, I'm wondering if the AKT pathway, does that also engage downstream mTOR signaling, for example? And if that's the case, do you look at like the cell morphology? Maybe they are bigger because the mTOR is related to that cell growth. Just want to see if you have any anatomical correlates of that.
Yeah. So I do. So I'm right now I'm looking at downstream kind of targets for the AKT pathway. mTOR was something that I kind of was starting to look at from the beginning, trying to determine. It's so complicated, to be honest. Like the mTOR pathway is gigantic.
Sure.
And so trying to like piece that apart is a whole separate project. But so I actually see, so not in the conditions that have the rescue, like the AKT drug. I haven't looked at that, but just in the SLC knockouts much later on. So I think I've taken them out to like 300, 500 days. And I do see some cells that have like a larger cell body. Like, I wouldn't say balloon cells, they're not balloon cells, but they have a much larger cell body. They just look really huge. And there's a few of them that are just kind of sprinkled throughout. I'm not really sure what those are. But yeah, we're still, the AKT pathway stuff still needs to be kind of pieced apart. Yeah.
Thank you.
Mm-hmm.
So thanks.
Hello.
Yeah, I was very interested in the migration effects that you reported. And you went to the oncology literature to look at the potential role of SLC6A1. There's also a lot of, I'm sure you know this, a lot of evidence for GABA itself being important for neuronal migration and maturation. So have you actually measured GABA in these in the wells of these cultures to see if the levels are changed?
So I haven't done HPLC or anything like that with the media, because I think with the diffusion of GABA and the organoid kind of gets a glial sheath around it when it starts maturing. So I'm not sure how much GABA would get out of the organoid itself, but I have done some staining of GABA in the organoids. And as far as I can tell with all the analysis we've done, it does not see that there's a difference in GABA, like just straight GABA at these early time points. There is, let me see what else would, I think there is some difference in GAD expression, like increased GAD expression. So maybe they're trying to, they're not able to uptake or reuse the GABA. So they're trying to make more. But yeah, that's as far as I've gotten with that.
Yeah.
So that's a great idea. Yeah, it's something that we've wanted to do, and I've kind of just kept putting it off because there's a lot of avenues to go with that.
Hey, I'd like to ask just one quick question. So, hey, this is Paul Carney, fantastic talk. So,
thank you.
So I love your MEA work. I've seen a few people leverage this. So my question is around the MEA and if you're planning to do any of the following. First of all, is this a 64-channel two-dimensional MEA or is it a 3D?
No, it's a 64-channel. We use the Axion system. It's a 64-channel 6-well MEA plate. I prefer the 6 wells. Other people
Question one: have you considered transitioning to a 4098 channel CMOS?
I would definitely if I had the opportunity.
Well, we can talk.
We've talked about high density MEAs in our lab, and I think the biggest barrier is like how are we going to deal with the massive amounts of data that it generates. So that's I think our biggest barrier right now.
That's my next question. So as Amber pointed out, seizures, we have not great seizure drugs. We can control hyperexcitability. I'm sure Jack can tell you all about that. Not great. Amber can tell me, but I don't know if this is a drug-resistant epilepsy. Someone else can tell me. But what I do know is they have these, I guess, behavioral, almost autistic-like, behavioral issues. So my question for you is, you have a really powerful technique to look at network. Have you gone beyond just looking at spike sorting and hyperexcitabilities? Have you applied any computational tools to see how that network evolves? From a communication, there's a lot of tools that you can look at. Have you considered that at all?
Yeah, we're actually, I mentioned him earlier and when I was talking about the EFIS, but we had a new postdoc that joined our lab and he is really digging into that. And he's an expert in EFIS. So he's digging into that, the whole maturation timeline of this stuff. So he's like really getting into it. So I think he's going to have a whole separate paper on that.
He's going to throw a lot of like communication tools, network tools, all this at those, is that what he's in? And then I think this company has a 3D. I know they have I think they have a beta now because we talked to them. They're actually developing a 3D, and then the group you may want to contact the group at Lawrence Livermore. They've actually developed a lot of that. Yeah, they're really good with Berkeley. Yeah, I can give you the name, but they're actually looking for collaborators to do exactly this kind of work. They've got this really nice 3D.
We're always willing to try 3D stuff because I think So the method that we
they're all engineers, so you're going to have to teach them the biology.
Yeah. We have some collaborators here at U of M that are also in the engineering department that are trying to do those kind of things.
You do.
I think everybody's trying to.
No, I'm a former
grad.
I'm a former graduate of UM. I did all my epilepsy
nice.
training. Yeah. So you're in the right place to do this work over in BME. So anyway, great.
Yeah.
Fantastic.
Yeah. The method that I developed for using this MEA is not the standard method where you plate the organoid onto the MEA and then basically the cells grow out and adhere, and then it kind of just becomes a 2D network at that point. But for what I've done, I've just kind of acutely plate them, remove the media so that it creates a little suction for the organoid onto the electrodes, and then you can gently add a little bit of media back and that suction will remain. And that gets us some really nice clean contacts with the electrode and really strong MEA data. And so then after you record it, you can just kind of blow some media off and the organoid can be removed and then reused again and again over time. And so you can actually track within the exact same organoid how that network develops.
Are you able to record in the during the incubation or no? Or you have to take them out?
Right now, we have to take them out and put them on the MEA plate acutely. Right. The MEA system that we use now has like an incubator, so you can leave them in there if you want. But yeah, it would be great. We're looking at a lot of optogenetic
we've
stuff
solved
right
that
now.
problem. We've kind of solved that problem. We can actually work. Yeah, we can record in the incubator without having to.
Nice.
Yeah. Anyway, but I'll stop there. Yeah. Fantastic.
Could you send me an email?
I will. Yeah, I'm so impressed with what you're doing. I can't believe this is a PhD. You sound like a faculty member, actually. The 3 IO1s.
I've been here many years. Thank you so much.
Okay.
Any other questions? Nope.
I will tell you, I moved back to Duke, so we're going to continue this work on a slightly different mutation called SCM1A, which is a presynaptic protein that's mutated. And I'm a pediatric neurologist. What's really interesting, all these syndromes, as you probably know, they all have very similar phenotypes. They seem to cluster. And we don't understand that, and this is really, and I think you have an opportunity to really make huge advances because the mouse is only going to get you so far. Yeah. Anyway.
Yeah. And Jack is actually, the lab also looks at SCN1A. So if you guys are interested in collaborating on a project with Jack when that, just reach out to him.
We are. And I should have asked it, and as we've heard this with Amber. One of the other things we're interested in, because I collaborate with UNC, is the heterogeneity, because you're looking at two humans, I think 2 cell types. But we know that there's too much heterogeneity, and what's the variability across the organoids of the stem cells, and how can we reconcile that? And then should we be looking at the mutation, or should we be looking at some of the modifier effects or the compensatory effects in development? Because you got this developmental thing going in the background, which many of us ignore, or at least some of us do. You know what I mean? Sort of, it's like it's like the drunk looking for the keys in the parking lot. Look where the light is. Well, it's not over there. It's somewhere else, right? So that's really the issue. And I think that has more legs for FDA. FDA actually asked me to come on as a reviewer, so I'm going to get some insight how they do this. And I do know that they are really focused on drugs big time. Okay, so if you can find a modifier, I loved your drug study. That was fantastic, by the way.
Yeah, and one of the nice things about the migration phenotype is that you can do large drug screens on not so much on the fusions, but when we can also plate them on just a thin coated gel tracks, and the cells will migrate out pretty rapidly. Like they start migrating out within like 2 hours. And so within 24 hours, you can get a whole lot of cells migrating out. And that's a nice readout for drug screening. And you could do really high density drug screening with that. And to get at your kind of variability question, that's one of the main reasons why we use the single rosette model, because it's just one rosette. And as far as cell types that are produced, it's very like homogeneous throughout. It's very, very reproducible, and they all look the same most of the time. When the variability comes in a little bit in the electrophysiological maturation timeline. So all the cells, all the organoids will have some kind of activity, but the duration of time that it takes them to reach like a network bursting, level can vary a little bit from organoid to organoid. So yeah, like I think most of them kind of start getting synchronous network bursting, I would say close to around 200, between like 180, 200. But there are some like that might come a little bit earlier, some cell lines. And so in our MGO study, we used like 3 different iPSC-derived lines, ones that are derived from fibroblast blood and from embryonic stem cells, and show that there's a little bit of difference in like the cell origin. So the ones that were fibroblast derived kind of developed their electrophysiological network properties a little bit later than the other 2. So it's important to have the same cell origin matched.
And you've gone with fibroblasts, not PBMCs, for strategic reason, right?
So I did both on my project. Yeah, I did the fibroblast and CRISPR edited them, and then I took the PBMCs from the patients. Yeah.
You're like one of the most thorough speakers I've seen on this topic. So anyway, thank you.
Thank you. Thank you.
I'm sorry. Thank you.
All right. Thank you.
Well, it sounds like it's time to start your fan club. We have now gone 20 minutes over. I think we could go way longer. This was amazing. Thank you.
Amber, I think you need to give best mock awards. Okay, we can all vote. Okay, with one of your baked goods. Okay. Yeah.
I do have good baked goods.
Just one thing, Amber, if anyone is interested in like delving in a little bit more to this MGO model, we do have it up on bioRxiv, and we're under revisions right now for that. So if anybody wants to look at that, it's on bioRxiv, and some of the SLC6A1 stuff is in there as well.
Okay, that's amazing. If you wouldn't mind sending me an email with the exact verbiage so I don't mess that up on the recap email I send out, I would really be appreciative.
Okay, sure. Thanks, Amber.
Can I ask one more question, or we're going to close it? I was going to, I just keep thinking, so calcium, right, as a second messenger is integral to that whole migration. You think the polarization of the GABA receptors, calcium transients, and then that causes all the downstream changes that improve motility. So I wonder if you could use calcium, like GCAP or something like that, to monitor how calcium dynamics are being affected. Maybe it's not sensitive enough, because really, I'm not sure if you're getting actual spikes. But then if you could, and I don't know if you did that, maybe you could do some real-time experiments, like block GAT1 with a drug and then see what happens in the wild type condition, or maybe try some sort of a rescue experiment. And then if it doesn't work, conclude that it has to maybe do with compensatory expression changes. You can't fix it real time, like KCC2 just goes up or something like that, but it kind of gives you another way to ask more questions. Do you think?
Yeah, for sure.
Possible?
Yeah, we actually, the other postdoc that I was telling you about, who's doing most of the EPHIS kind of characterization stuff, he's also doing a lot of calcium imaging, just characterizing them in our MGOs over time, developmentally and everything like that. And so, yeah, we can see like spikes, even early on, we can see really nice calcium spikes, even like 40 days. There's some like, here and there, not network wide, but there is definitely activity. And so it would be great to like look at that with SLC6A1. We just kind of got started on the calcium imaging train in the lab. So it's still in kind of early as far as implementing that in SLC6A1, because I'm like done. So it's stuff that Jack will definitely probably follow up on. And so, yeah, it would be great to look at that and test some drugs. We also have some really interesting, there was a paper that came out with interneuron subtype specific, like viral labeling. You can do optogenetics on them, targeting specific interneurons. So we have like the basket cell targeting, the chandelier cell targeting, and we've been working with that so that we can specifically target, like if we're interested in, is it more the proalbumin cells that are regulating this hyperexcitable phenotype? We can kind of perturb just those cell types, just that cell type, maybe just somatostatin, maybe just the basket cells. So that's also something that our postdoc is working on right now.
Yeah. How long has your PhD been? I'm curious.
I've been here 7 years, so I really shouldn't have like the first project, the MGO project, was supposed to be the low-hanging fruit. It was supposed to be an easy, like, hey, yeah, just make this, and then you can do the rest. But that actually ended up taking 6 years in itself to do. And along the same time, I was doing my SLC stuff. So
have you
yeah.
published any of this?
So it's on bioRxiv right now, and we're
I'll take
under
a look.
revision.
Perfect.
So fingers crossed, hopefully it'll be accepted soon.
So everything you presented is in public domain today, right?
Not everything, no.
Okay.
Some of the SLC data is not out there yet. It will be a part of another publication that I'm gonna put out. So hopefully soon. I'll
Great.
put it up on bioRxiv once I get it submitted. Yeah.
And I have one more question. I'll stop. Amber, this migration is really quite impressive. This whole migrational theme or hypothesis. Is anybody doing like really advanced brain imaging to try to phenotype the structure? Do you know? I don't mean just like 3T. Other there's like structure and migration or deep migration or dysplastic lesions, if you will, is really a hot topic because it's considered the most important structure abnormality for tractable epilepsy. Okay, so 7T imaging and other kinds of ways. There's even ways to do this very quickly, like with fingerprint imaging. Do you know if anybody's doing any of this to look at the structure and correlate that with the phenotype?
Would any of that fall under a MEG or an MRS?
MRI, structural MRI. NIH has funded several really in recent years. I'm on the committees. I can tell you, I've seen these. I'm looking at advanced techniques to look at structure, and people have taken that and correlated with phenotype because what we're seeing here today is this is a really important area, and there'll be some. I'm just curious if anyone's doing anything like this. Do you know?
We have an MRS study at UT Southwestern. If David's still on, he can speak to that. Scott Perry at Cooks is doing a MEG study, but that's not necessarily SLC6A1. But
I've heard no names of really kind of people just work on imaging. These are clinicians like me. Who work in the topic. So MRS is looking at peaks, chemistry, and the other one, MEG, is looking at, epileptiform discharges, but which would be kind of function. I'm talking about structure. And so, because we know, because the structure defines some of the phenotypes, depending where it's at. Okay, prefrontal cortex, hippocampus, so forth. So, yeah, I can send you some names of people I know would be really interested. Actually, where I'm at now in the RTP, there's really a lot of good places, but this is an area, if you just tap into the epilepsy world where people with epilepsy surgery, for example, this is really where they spend most of their time looking for structural. And then there's structure function, right? So, structure shapes function, but it doesn't define it. So this is kind of where people look at. But what we've seen today is the whole migration. I think that's critical. Anyway, but I'll follow up with you, give you some names you might want to reach out, or I can reach out for you because you have such a great team here of international people. People will be very interested in looking, and we don't need a lot of subjects. We can get travel grants and grants to kind of do the same. I'll talk to you.
Okay. All righty. Well, that was one heck of a meeting.
Yeah, great presentation.
Sorry we ran over so much.
Everybody have a great Thursday and a happy Valentine's Day.
Thank you. Bye.
Bye.